we're able to find out the genetic basis of how we can grow more out of a limited land capacity, we basically can fight a very big problem. Food, nutraceuticals. Everybody wants to get up in the morning and take their vitamins, take their omega-3 fatty acids and so on. These kind of processes are not possible without an understanding of how to culture microbial organisms in large reactors where they can basically engineer these organisms to produce things like vitamins and fatty acids and so on. So, genomics is really the study of genes and genomes. It's really kind of like how you would say a branch of genetics, but focused on what the genome is actually doing. And it can extend to things like plant cells, animal cells, depending on what area. I mean, you have people who are working in different industries here, as I mentioned earlier on. So we have about, let's say we have 1,000,000 proteins, right? And each gene and protein can have different forms, and these are like isoforms. You might have a gene which can you might code for about 5 or 6 splice forms, and each splice form may be in a different part of the body or the cell. And we can't really exactly tell without expression information what cells are expressing which isoforms. And also, we have, to further complicate the problem, each gene and protein can interact with multiple partners. We have transcriptional regulation. We have things going on at the nucleus, in the nucleus near the DNA. We have transcription factors interacting with each other. Quantity is another problem. Each gene or protein can be made in different amounts and places in the body. I mean, we need to be able to quantify these things with different experiments. People are doing microarrays, people are doing sequencing genomes. And basically, these are all compounded problems where we basically have to kind of, sometimes we have to be reductionist in looking at the problem in a very simple manner. Sometimes we have to design more complex algorithms to mimic multiple interactions and so on. So what it leads us to is we have many permutations, and basically, if you calculate that, it probably gives you the number of atoms in the universe, which is a crazy amount. So in genomics, bioinformatics, there's too much data to analyze. And using, if we sat with I think I was at a presentation with someone from SIB in Switzerland, and he said that, if you wanted to analyze like the human genome, you need more paper